name is Hana Marvanová. I am a PhD student at IMBA, the Institute of Molecular Biotechnology in Vienna. I work on parent of origin effect, and I use Senrabdetis elegans, a little worm, to actually investigate that topic. I grew up in Prague. I had quite average and happy childhood. There is a story though. When I was 4 years old, my mom brought me to the Natural History Museum in Prague, and unexpectedly I couldn't be pulled away from the skeletal exhibits of the early hominins there. She probably wanted to show me some butterflies or something very nice and colorful, and yet I was staring at bones. And this interest in human kind of persisted for me, and that's what made me study anthropology at the Charles University in Prague. And then the story was quite, it took some turns and it was quite long, but I ended up here doing something completely different. Well, it's quite important because it's the bridge between Africa and Europe. And the anatomically modern human was actually emerged in Africa. And afterwards, this species spread across the rest of the world and eventually dominated it. And in the lab of archaeogenetics, they are doing an amazing job where they compare the DNA from saliva samples from different populations in Africa, and that allows them to track the steps of the anatomically modern human 100,000 years ago to decipher precisely the road that this human took to spread from Africa. And I think that's just wonderful. On the research level, I think there is an umbrella interest of mine, which is the origin of human, but from different perspectives. So when I worked in Prague, I was interested in evolution of human, which can answer some of the questions that I have regarding who we are. But also another interesting perspective is the early human development and basically fertilization and the early development of ours. And that's what brought me to the Max Planck Institute, where we studied gametes or sperm or sex cells, one could say. And we also studied selfish genetic elements that are responsible for a lot in our evolution because they are driving the genetic conflict, which is the basis for the evolution. They're all about human, in a way. They tell us more about how are we shaped. The archaeogenetics, for instance, how did the environment of the early anatomic human look like, tells us more about our today's society. And then the topics that I have researched in Max Planck and here at BBC tell us more about the evolution and the genomic conflict that is responsible for evolution, that focuses on kind of different origin, but it's still the origin of our species in a way. Before I boast, I am only a second author on this paper, okay? So, but my colleagues did a wonderful job in bringing this story to life. So, very briefly, we inherit the complete set of genes in form of chromosomes from our mother and from our father. That means that we have two versions of each gene in our bodies available. Now, that doesn't mean that each of them gets utilized equally. Sometimes one of these versions gets ignored, silenced, and only the other one is used. That's something that we call the parent of origin effect. We have examples of parent of origin effect also in human. We even have diseases that are caused by parent of origin effect. we don't know how it evolved, and we also have only theories why it evolved. In the beginning of this research, there was a theory saying that these differences occurred because of different interests of the mother and the father. But that still doesn't tell us how they actually emerged. And one theory says that it evolved from the DNA defense mechanisms that are brought to action by the piRNA pathway. But the connection between these two was missing until these days. We were lucky enough to discover a case of a parent of origin effect in the nematode Caenorhabditis tropicalis that actually helped us to discover the missing piece between this defense mechanism and the parent of origin effect. We work quite closely together. We divide the tasks. We frequently discuss what is important for the story, and there is always the group leader who kind of has in mind how the story should continue, what is the next important question that needs to be answered immediately. And then we just divide the tasks and divide the immediate experiments, the next important experiment that needs to be done based on our previous experiences. So if I have a colleague who is good at doing a Western blot, I will simply ask him to do that because it's the best for the quick progress of the project. There are many of these questions that I'm not sure if they can be answered, but they should definitely be discussed. As I kind of incline towards infertility research, of course, these questions of designer babies or in vitro fertilization techniques are kind of closer to my interest, and I pay more attention to these questions. But of course, there is always the gene editing or the data protection. As genome sequencing gets cheaper and cheaper, and it's probably going to get used quite soon. So all of these topics, I believe, should be taught and should be discussed by scientists, because it definitely clarifies that we need to communicate our science better, because we need a society that is interested and informed and is willing to bring attention to the new legislation that we will need to have for these upcoming progresses. This is the largest scientific hub I have worked at, and that has multiple benefits. I personally feel quite spoiled here, given how many shared facilities we have at hand just for us to use. But more importantly, because so many scientists are already here, many scientific conferences take place in Vienna every year. And that means the experts of the respective fields are coming to town. And then it's quite easy for PhD students of Vienna to actually meet their heroes without even traveling. And that's something absolutely amazing, I think. Absolutely. Even on the emotional level, if you can shake hands with your hero, with somebody who just started a new field that you are currently working at, it's amazing. The first example that comes to my mind is AlphaFold, which was a struggle for scientists working with proteins for last 20-30 years. It was believed that it's impossible to predict a structure of a protein just based on the code sequence. AlphaFold basically solved that for us. There was Nobel Prize for that already awarded. So that was like the big shake of the last years for our field, because with these predictions of protein structures, everything is much quicker for us, even if we, like for instance in my case, don't directly work with proteins, but some related questions. Right now, I focus on finishing my PhD And that's still gonna take some time. But I got training in handling multiple gametes, sex cells of multiple different organisms, including human. And in the future, I would like to contribute to the research of infertility and hopefully enhance some of the in vitro fertilization methods, for instance. That's my scientific goal for the next chapter, let's say. Besides that, there is still the anthropologist in me who would really love to find new ways how to introduce scientists as a sort of people to the public, because I believe they make really great role models. We just need to introduce them as cool as they are, you know. I would congratulate them on the curiosity that they have. I would advise them to follow it, because curiosity is the best thing you can have, especially in science, but generally speaking. But I would also tell them to pay attention to the choice of people and environment that they work at, because having fun and having supportive environment is crucial in research. It's so much easier when you have supportive people around you.